Synthetic pyrethroids are synthesized derivatives of naturally occurring pyrethrins, which are taken from pyrethrum, the oleous resin extract of dried chrysanthemum flowers. The insecticidal properties of pyrethrins are derived from ketoalcoholic esters of chrysanthemic and pyrethroic acids. These acids are strongly lipophilic and rapidly penetrate many insects and paralyze their nerve system. Mode of action: pyrethroids are axonic excitotoxins and toxic effects of which are mediated through preventing the closure of the voltage-gated sodium channels in axonal membranes. The sodium channels is a membrane protein with a hydrophilic interior. This interior is shaped precisely to allow sodium ions to pass through the membrane, enter the axon, and propagate an action potential. When the toxin keeps the channel in their open state, the nerves cannot repolarize, leaving the axonal membrane permanently depolarized, thus paralyzing the organism. Pyrethroids can be combined with the synergist pepperoyl butoxide, a non-inhibitor of key microsomal cytochrome P450 enzymes from metabolizing the pyrethroid, which increases its efficiency or lethality. It is likely that there are other mechanisms of intoxication also. Disruption of neuroendocrine activity is thought to contribute to their irreversible effects on insects, which indicate a pyrethroid action on voltage-gated calcium channels and perhaps other voltage-gated channels more widely. Alethrin. Alethrin was the first synthetic analog of pyrethrum developed in 1947 by Green and Laforge. Alethrin is a common name for RS3 allyl 2-methyl 4-oxocyclopentane 2-anyl IRS cis trans chrysanthemate. It was first discovered by M.S. Schertz et al. In 1949 and introduced by Sumitomo Chemical Company. It has contact, stomach, and respiratory action and brings about quick knockdown of flies and mosquitoes when applied in combination with synergists like piperonyl butoxide. The acute oral LD54 rat is short pyrethrin A. Pyrethrins are powerful contact insecticides but appear to be poor stomach poisons. A characteristic action of pyrethroid is the rapid paralysis of knockdown effect and substantial recovery from that. So this discovery is due to rapid enzymatic detoxification in the insect. To bring about a mortality equivalent to the knockdown effect, about 3-fold increase in dosage may be required. Compounds such as piperonyl butoxide, propyl isome and sulfoxide are known to inhibit the detoxification enzymes and increase the toxicity of pyrethroid. Pyrocone. One part of pyrethrin and parts of piperonyl butoxide is used for the control of coconut red palm weevil. Pyrocolloid, a colloidal form at 1 in 400 to 600 parts of water, is useful against aileroidids and scales in household sprays and as a repellent against external parasites of livestock. Pyrethrum is useful. It is also mixed with stored grains for control of stored grain pests. Pests of coconut. Rhinoceros beetle, Oryctes rhinoceros, Scarabaeidae, Coleoptera, host range pineapple, sugarcane, arecanut, sago, oil palm, palmyra, date palm, and wild dates. Damage symptoms: central spindle appears cut or toppled. Fully opened fronts show characteristic diamond-shaped cuttings. Holes with chewed fibers sticking at the base of central spindle. Bionomics: female lays up to 140 to 140 oval creamy white eggs in manure pits or decaying vegetable matter at a depth of 5 to 15 cm. Egg period 8 to 18 days. Stout sluggish white grub with pale brown head is found at a depth of 5 to 30 cm. Grubs feed on the decaying matter and grub stage lasts for 99 to 182 days. Grub pupates in earthen cells at a depth of 1.3 to 1 m. Damage symptoms: central spindle appears cut or toppled. Fully opened fronts show characteristic diamond-shaped cuttings. Then holes with chewed fibers sticking at the base of central spindle. Bionomics: female lays up to 140 oval creamy white eggs in manure pits or decaying vegetable matter at a depth of 5 to 15 centimeter. Egg period 8 to 18 days. Stout sluggish white grub with pale brown head is found at a depth of 5 to 30 centimeter. Grubs feed on the decaying matter and grub stage lasts for 99 to 182 days. Grub pupates in earthen cells at a depth of 0.3 to 1 meter and emerges as adults in 10 to 25 days. Adult beetle is stout, black and has a long horn projecting dorsally from the head in male. Horn is short in female. Horn is short in female. Control: destroy and dispose all dead trees. Avoid manure pits in the vicinity of coconut gardens. Rake and turn up the decaying manure to expose the developing grub and egg and pupae to sun drying and predation. Then apply the fungal culture of Metarhizium anisopliae to manure pits during cooler months of October-December. Encourage reduced parasites Platymerus laevicolus. Once in 3 months, drench the manure pits with carbaryl. In seedlings, place naphthalene balls, 3 per tree, in the innermost 3 leaf axils once in 45 days. So in seedlings, place naphthalene balls 3 per tree in the innermost 3 leaf axils once in 45 days. Soak castor cake 1 kilogram per 5 liter of water in wide-mouthed mud pots and keep them in a garden to attract and kill adults. Replace the slurry once in 30 days. Fermented toddy may be kept in wide-mouthed earthen vessels in different places to attract the adults during night. The crown region may be poorly cleaned during harvest and adults may be hooked out using a long wire. Light traps may be set up to attract the adults during monsoon months and following rains during summer. The topmost 3 axils may be filled with a mixture of sand plus neem seed powder once in 3 months. Use aggregation pheromone traps. Rhinolure. Install the trap at 5 feet from ground level. Red palm weevil, Rhynchophorus ferruginous. Distribution and status: Kerala, Karnataka, Tamil Nadu, Assam and Maharashtra enjoy major pest status. Then damage symptoms: holes on the trunk with brownish ooze, yellowing of inner leaves and gradual wilting of central shoot in crown. Up to 5 number of grubs can be found feeding on the soft tissue inside the trunk. Damage symptoms: holes on the trunk with brownish ooze, yellowing of inner leaves and gradual wilting of central shoot in the crown. Up to 5 adenoids of grubs can be found feeding on the soft tissue inside the trunk. Life cycle: egg female lays up to 276 oval white eggs in the scooped out small cavities of palms or up to 7 years. And on older trees, it deposits the hands and other cut injuries of trunk. Egg period 2 to 5 days. Grub apodus Light yellowish grub with a red head becomes full grown in 36 to 78 days and pupates in a fibrous cocoon inside the trunk itself. Reddish brown adult weevil has 6 dark spots on thorax. Male has a conspicuous long snout with tuft of hairs. Control. Remove and disposal of damaged and wilted trees. Avoid injuries on trunk and any injury should be plastered with clay or cemented with copper oxychloride. Avoid cutting green fronds. Root feeding with monocrotophos. 10 ml water should be done after harvest of nuts. Observe a waiting period of 45 days. Set up attractant traps using mud pots with molasses, toddy 2.5 liter, acetic acid, molasses or toddy 2.5 liter, acetic acid 5 ml, yeast 5 grams, split tender coconut stems, petioles. Then insert 1 to 2 aluminum phosphate tablets inside the tunnel and plug all the holes with clay plus copper oxychloride. Use aggregation pheromone traps. Ferrolure in combination with food baits consisting of 1 kilogram sugarcane molasses, 1 gram of yeast, 5 gram of yeast, 1 ml glacial acetic acid, split petioles of coconut taken in a bucket of 10 liter capacity. Black-headed caterpillar. Black-headed caterpillar. Opicina arenocella. Cryptophacidae Lepidoptera. Distribution and status all over Peninsula and India, east and west coasts. Damage symptoms: dried up patches on leaflets of the lower leaves, galleries of silk and frass on underside of leaflets. Black-headed caterpillar, grayish-white small mouth, lays about 180 eggs in grounds on leaves. Egg period is 5 days. Greenish brown larva with dark brown head and prothorax and a reddish mesothorax. Larval period 40 days, pupal period 12 days. It pupates inside the web in a thin silicon cocoon. Control: cutting and burning all the infested leaves and fronds. In small plantations, carbaryl may be sprayed. In summer, release bathylids, braconid and eulopid parasitoids from January. Root feeding with monocrotophos. 10 ml plus 10 ml water with a waiting period of 45 days after root feeding. Calories of silk and frass to underside of leaves. White grub. Leucophilus coniophora. Melanolonidae coleoptera. Host range: sweet potato, tapioca, colocasia and banana, raised as intercrop in coconut plantations. Leaves turn yellow, immature nuts shed, flowering delayed. White grub was exposed when base of the tree is dug. Life cycle: female lays eggs in a soil at a depth of 7 to 15 centimeter. Egg period is 20 days. Grub period is 10 to 11 months. Pre-pupal period is 9 to 12 days. Pupal period is 25 days. Pupation occurs in soil. Adult beetle emerges after monsoon showers. Control: summer ploughing exposes the immature stages. Sow the crop early in the Kharif season. Treat the seeds with chloropyrifos 12 ml per kilogram of kernels. Apply 4-8 or carbofuran in the soil at or before sowing. Spray 500 gram carbaryl on the preferred hosts like guava, banana and in the vicinity. White grub. Lace wing bug. Stephanotis typicus. Symptoms of damage: white spots on the upper surface of the leaves. Lace bug. sap feeds from coconut foliage. It acts as a vector in transmission of phytoplasma from root affected palms to healthy palm. Identification of the pest: nymph white with dark patches, adults white colored, adults with netted venation or wing. Control method: remove leaflets harboring these insects and destroy them. Chemical method: spray any one of the following: malathion dimethoate, methyl dimethoate, phosphyl amide, metomyl 3%, neem oil. Coconut skipper. Pest population occurs around the year, but population maximum during June September, coinciding with the onset of monsoon. Then symptoms of damage: one half of the leaflets are cut and rolled into a case. The rolled leaflets are dried. Coconut skipper. Control chemical method: collect and destroy the immature stages of the insect by conducting study or neem campaign whenever possible, and spray carbaryl 50 WP 2 gram per liter. Spray dichlorvos. So this is about coconut pests. Rhinoceros beetle, then a red palm weevil, black-headed caterpillar, white grub, lacewing bug, coconut skipper. Yeah. Pesticides and environment. Next topic. The environmental effects of pesticides describe the broad series of consequences of using pesticides. Pesticides, because they are toxic chemicals meant to kill pest species, can affect non-target species such as plants, animals, and humans. Over 98% of sprayed insecticides and 95% of herbicides reach a destination other than their target species because they are sprayed or spread across entire agricultural fields. Other agrochemicals such as fertilizers can also have negative effect on the environment. The negative effects of pesticides are not just in the area of application. Runoff and pesticide drift can carry pesticides into distinct aquatic environments or other fields, grazing areas, human settlements, and undeveloped areas. Other problems emerge from poor production, transport, storage, and disposal practices. Over time, repeat applications of pesticides increase pest resistance, while its effect on other species can facilitate the pest's resurgence. Alternatives to heavy use of pesticides such as integrated pest management or sustainable agricultural techniques such as polyculture mitigate these consequences without the harmful toxic chemical application. A farm worker wearing protective equipment pouring concentrated pesticide into a sprayer tank with water. Drainage of fertilizer and pesticide into stream. Pesticides being sprayed into a recently plowed field by tractor. Pesticide impact on wildlife in soil. Pesticides reach soil as a result of direct application to it or by drift during dusting and spraying or drifting and dusting and spraying to foliage or washed down by rains after application. From soil, the persistent chemicals enters into soil invertebrates of water and are broken down by microorganisms and physical factors. Such chemicals are mostly DDT and dieldrin. Aldrin also breaks down in soil to dieldrin. DDT and dieldrin are reported to be most prominent soil contaminants in Western countries, perhaps because of their large scale and indiscriminate use. The persistence of chemical in soil is influenced by various factors. DDT and the persistence of chemicals in soil is influenced by various factors like the granular formulations persist longer than emulsions, which again persist longer than vegetable powders. Heavy clay soil retain pesticides longer than light sandy ones, and both the organochlorines and organophosphates persist longer in acid than in alkaline soils. Higher into organic matter and clay mineral content in a soil, longer is the persistence of a chemical. Higher the temperature and soil moisture, less is the persistence. And then the breakdown of insecticide is more in soil having microorganisms than in sterile soils. In water, pesticides reach water in lakes, ponds, tanks, rivers, and streams from application of the same in mosquito control deposits from aerial spraying of cultivated crops or a surface runoff from soil treated with the chemical from disposals and wash off of insecticide contaminants, etc. Because of the absorption of the chemicals by bottom, sand, mud, planktons, aquatic plants and invertebrates and of the breakdown due to hydrolysis, toxic concentrations in water seldom build up. However, the two main hazards of residues in water are the deposition of chemicals in the bodies of fishes because of the absorption of the chemicals by bottom, sand, mud, planktons, aquatic plants, and invertebrates are of breakdown due to hydrolysis. Toxic concentrates in water seldom build up. However, the two main hazards of residues in water are the deposition of chemicals in bodies of fishes, which are effective fillers of suspended particulate matter, and in the bodies of aquatic organisms that form the food for fish. And as a result of both these, more chemical agents deposited in the fishes than is found free in water. The accumulation of pesticides in fishes is of great importance because they are food for many animals and birds and man. In biota, plants retain pesticides in their surface or in tissues as a result of chemical control of pests. Organophosphorus compounds and systemic poisons are soon broken down, but organochlorines like dieldrin are retained for relatively long period. It is important that crops used for human or animal food should not contain residues of pesticides, and hence tolerance levels that can be permitted in crops used for food have been five for most chemicals. In birds, residues of organochlorines occurs in tissues and eggs of many species of birds, and these residues might have accumulated from its food, mainly from fish and also from grains, leaves, etc. The birds accumulating lethal dose of poison may get killed, followed by any predatory and scavenger birds feeding upon them. It has also been reported that residues of organochlorines in their tissues lower the vitality of chicks and eggs. However, birds lose the chemicals when they are no longer exposed to them. In food, pollution is through application of persistent pesticides just before the harvest or during storage. The important groups of chemicals that tend to persist in plant tissues and are transferred to man and animals include DDT, HCH, and dieldrin. In the body of animals, they get accumulated in fat tissues and are present in milk and dairy products. Exposure can be linked to cancer, endocrine disruptions, reproductive effects, neurotoxicity, kidney and liver damage, birth defects, and developmental changes in a wide range of species. And exposure to pesticides can also alter an organism's behavior, impacting its ability to survive. In birds, for example, exposure to certain pesticides can impede singing ability, making it difficult to attract mates and reproduce. Then, pesticides can also affect birds' ability to care for offspring, causing their young to die. For bees, even near infinitesimal levels of systemic pesticides result in sublethal effects impacting mobility, feeding behavior, and navigation. Many deformations have been found after exposure to hormone mimicking pesticides classified endocrine disruptors. Impact of these chemicals include hermaphroditic deformations in frogs, pseudo hermaphrodite polar bears with penis like stems, and panthers with atrophied testicles, and intersex fish and rivers throughout the US. Reproductive abnormalities have been observed in mammals, birds, reptiles, fish, and mollusks at exposure levels considered safe by the US Environmental Protection Agency (EP). Impact on human health. Acute immediate health effects. Immediate health effects from pesticide exposure include irritation of the nose, throat, and skin causing burning, tying and itching, as well as rashes and blisters. Nausea, dizziness, and diarrhea are also very common. People with asthma may have very severe reactions to some pesticides, particularly pyrethrin or pyrethroid, organophosphate, and carbamate pesticides. In many cars, symptoms of pesticide poisoning mimic symptoms of cold, and a flu. Since pesticide-related illness appear similar and identical to other illness, pesticide poisoning are often misdiagnosed or underreported. Immediately, symptoms may not be severe enough to prompt an individual to seek medical attention, or a doctor might not even think of ask about pesticide exposure. Still, seek medical attention immediately if you think you have been poisoned by insecticides. Chronic, ionic, long-term health effects. Chronic health effects include cancer and other tumors, brain and nerve system damage, birth defects, infertility, and other reproductive problems, and damage to the liver, kidney, lungs, and other body organs. Chronic effects may not appear for weeks, months, or even years after exposure, making it difficult to link health impacts to pesticides. Pesticides have been implicated in human studies of leukemia, lymphoma, and cancers of the brain, breasts, prostate, testicles, and ovaries. Reproductive harm from pesticides includes birth defects, stillbirth, spontaneous abortion, sterility, and infertility. Endocrine disruptors are chemicals that often at extremely low doses interfere with important bodily functions by mimicking or blocking hormones, the chemical messengers that circulate in blood and regulate many body processes include metabolism, brain development, sleep cycle, and stress response. Some pesticides act as endocrine disruptors and have been shown to cause serious harm to animals, including cancer, sterility, and developmental problems. Similar impacts have been associated with human exposure to these chemicals. Allergic effects. Allergic effects are harmful and effects that some people develop in reaction to substance that do not cause the same reaction in most other people. Types of allergic effects. Some people are sensitized to certain pesticides after being exposed once or a few times without effect. They develop a severe allergy-like response upon later exposure. These allergic effects include systemic effects such as asthma or even life-threatening shock, skin irritation such as rash, blisters, or open sores, and eyes and nose irritation such as itchy, watery eyes, and sneezing. Yeah. Insecticide resistance, genetic, physiological, and biochemical mechanism. Next topic. The objective of pest management practice is to lower pest status by reducing pest number and damage potential. This is well and good, but this objective is not always achieved. Ecological backlash involved the counter response of pest population or other biotic factors on the environment that diminish the effectiveness of management tactics. An important feature of the objective of pest management practice is to lower pest status by reducing pest number and damage potential. That is well and good, but this objective is not always achieved. Ecological backlash involves the counter responses of pest population or other biotic factors on environment that diminish the effectiveness of management tactics. An important feature of ecological backlash is that it is usually delayed. Ecological backlash has occurred with shocking frequency in the history of pest management. An understanding of ecological backlash rests on a knowledge of the major sources of phenomenon. These sources can be called the 3 R's of pest management awareness. These are population resistance, resurgence, and replacement. Population resistance, resurgence, and replacement. Insecticide resistance. Insecticide resistance is a heritable change in the susceptibility of a pest population to a particular insecticide or insecticide group. In practice, insects that repeatedly fail to be adequately controlled by the registered rate of an insecticide are said to be insecticide resistant. For insecticide resistant levels are high, pest population may be unaffected by many times and normal rate of insecticide application. The development of an ability in strain of insects to tolerate doses of toxicants which would prove lethal to majority of individuals in normal population of the same species. Resistance is quick to develop to compounds with high effective kill, long residual, and highly selective at a single biochemical target site. The toxicant is converted into a non-toxicant form in the body of insect by various enzymes. All these enzymatic changes are carried forward and transmitted through genes, single or multiple. A knowledge of insecticide resistance is important from point of view of proper selection of insecticides. Natural selection in action, pesticide resistance. Before parasite application, first generation. After parasite application, later generation, more resistant individuals are there. Resistant insects may detoxify or destroy the toxin faster than susceptible insects or prevent the toxin from reaching target sites by binding it to proteins in their bodies. Metabolic resistance is most common mechanism and often represent the greatest challenge. Resistant insects may possess higher levels or more efficient forms of enzymes that break down insecticides to non-toxic compounds. Examples: studies in America have shown that fruit flies have infested orange groves were becoming resistant to malathion. In Hawaii, Japan, and Chinese and diamondback moth evolved a resistance to Bacillus thuringiensis about 3 years after it began to be used heavily. DDT is no longer effective in preventing malaria in some places. Resistance developed slowly in the 1960s due to agricultural use. In the southern United States, Amaranthus palmeri, which interfere with cotton production, has evolved resistance to the herbicide glyphosate. glyphosate. Multiple resistance pests are resistant to more than one class of pesticide. This can happen when pesticides are used in sequence and with a new class replacing one to which pests display resistance with other. So you are learning about multiple resistance. Multiple resistance pests are resistant to more than one class of pesticide. This can happen when pesticides are used in sequence with a new class replacing one to which pests display resistance with another. Cross resistance, a related phenomenon, occurs when the genetic mutation that made the pest resistant to one pesticide also makes it resistant to others, often those with a similar mechanism of action. Metabolic resistance, resistant insects may detoxify or destroy the toxin faster than susceptible insects or prevent the toxin from reaching target sites by binding it to proteins in their bodies. A related phenomenon occurs when the genetic mutation that made the pest resistant to one pesticide also makes it resistant to others, often with similar mechanism of action. Then metabolic resistance. Resistant insects may detoxify or destroy the toxin faster than susceptible insects to prevent the toxin from reaching target sites by binding it to toxins in their bodies. Metabolic resistance is the most common mechanism and often presents the greatest challenge. Resistant insects may possess higher levels of more efficient forms of their enzymes that break down insecticides to non-toxic compounds. Altered target site resistance. The site where toxin usually binds in insect become modified to reduce the insecticide's effect. Behavioral resistance. Resistant insects may avoid the toxin by a change in their normal activity. Insects may simply stop feeding or move to the underside of a sprayed leaf. Some malaria transmitting mosquitoes in Africa developed a preference for resting outside that prevented them from coming in contact with the pesticide sprayed on interior walls. Then penetration resistance. Resistant insect may absorb the toxin more slowly than susceptible insects. Penetration resistance occurs when the insect's outer cuticle develops barriers that can slow the absorption of the chemicals into their bodies. This mechanism is frequently present along with other types. Genetic resistance mechanism. Genetic resistance occur naturally with any population and can be transferred from one generation to the next. Continued use of insecticide or insecticide group with common mode of action over several generations will select for resistance. While the insecticides will kill most pests, a small minority will survive and multiply, and this proportion of survivors increase with each generation. Resistance take while to develop, such as R alleles are initially rare. The rarity of R allele suggests it comes with reduced fitness. Thus, R allele should fall to a very low frequency without insecticide selection. Genetic factors. Genetic factors, number of R gene, quicker if one major gene, dominance of R allele, RR quicker if R allele is dominant, the initial frequency of R allele, the greater the quicker or the higher the quicker, fitness of R allele, quicker if no fitness cost. Plant breeding activities aimed at developing resistance rely heavily on knowledge of genetic background for the resistance. Such knowledge provide a quantitative basis for designs to recombine genes and select of proper characters. It also allows the identification of stable resistant factors that are least likely to be overcome by a pest population. Genetic insecticide resistant mechanism can be broadly classified in four major groups. Point mutations in insecticide targeted genes. Overexpression or mutations in the coding region of genes that code for detoxification enzymes. Overexpression or mutation in genes involved in cuticle formation. The gene for gene relationship. So, genetic insecticide resistant mechanism can be broadly classified in four major groups. Point mutation in insecticide targeted genes, overexpression or mutation in the coding region of genes that code for detoxification enzymes, overexpression of mutations in genes involved in cuticle formation, the gene for gene relationship. The gene for gene relationship. Many pest population include individuals with virulent genes, which allow a pest species to overcome resistance and once more attack a plant. One or more virulent genes may be present that allow an individual pest to overcome the effects of one or more plant genes responsible for resistance. This principle has been called the gene for gene relationship. Once again, many pest population include individuals with virulent genes, which allow a pest species to overcome resistance and once more attack a plant. Once one or more virulent genes may be present that allow an individual pest to overcome the effects of one or more plant genes responsible for resistance. This principle has been called a gene for gene relationship. In gene for gene relationship, plant cultivars are resistant because they have a resistant allele at a gene locus that correspond to an virulent allele at an equivalent locus in the insects. Judging from the range of effectiveness of a resistant plant variety, Planck recognized two types of resistance, vertical and horizontal. In etymological terms, vertical resistance refers to cultivars with resistance limited to one or a few pest genotypes. Horizontal resistance distribute cultivars that express resistance against a broad range of phenotypes. Arguments against vertical resistance in breeding programs because the potential development of biotypes, but it has been successful in many instances that Haitian fly on wheat, and it is easier to incorporate into new varieties than horizontal resistance. To manage to manage insect by using vertical resistance, it has been suggested that resistant plant genes need to be identified and incorporated into varieties that are held, then released when biotypes appear. Horizontal resistance has low heritability and is difficult for plant breeders to incorporate. Horizontal resistance may be the most desirable type of resistance to use in pest management because its stability. Physiological mechanism of resistance. The biochemical and physiological mechanisms of insecticide resistance are known to evolve along several trajectories. Behavioral resistance and decreased cuticular penetration allows insect avoiding lethal effects of insecticides as they come in contact with or consume insecticides. Once the insecticides enter the organism, enhanced metabolic detoxification, sequestration, or excretion of the toxins may take place in resistant insects before the insecticides ever reach their target site. Resistant insects are also likely to evolve target site insensitivity mechanisms which reduce or eliminate the binding affinity of insecticides to their target proteins. Commonly, a phenomenon of insecticide resistance is that these mechanisms operate simultaneously in resistant insect populations to contribute to overall resistance. Whether soil, plant architecture, and cultural practices are some of the most important influences on plants' physical environment. These factors can affect plant resistance by influencing such elements such as temperature, light intensity, and soil fertility. Changes in these elements cause fundamental change in plant physiological processes and can alter levels of allelochemics or cause imbalance in basic nutrients. Temperature. Abnormally high or low temperature for a period of time may cause loss of resistance. As an example, exceptionally low temperature have caused the loss of resistance of some alfalfa genotypes. So, spotted alfalfa aphid and pea aphids and some of the sorghum genotype to greenbug. Loss of resistance to Hessian fly has been found in some wheat and at temperature above 150 degrees Celsius. Light intensity. Shade induced loss of resistance has been found in several instances. These include wheat resistance to the wheat stem sawfly, Cephus cinctus, sugar beetles, resistant to green pH aphid, Mysis persistent, Persial and potatoes resistant to the Colorado potato beetle. With potatoes, shading was found to be associated with reduced feeding and development of the beetle. Soil fertility, changes in soil nutrients. Temperature, light intensity. Changes in soil nutrient level may also mediate the expression of resistance in some plants, but little is known about the mechanism involved. In an example of this phenomenon, we mentioned resistance are increased by deficiency. Clones of alfalfa resistant to the spotted alfalfa aphids were found to be reduced resistance if deficient level of calcium or potassium or excess level of magnesium or nitrogen were present. In the same example, resistance was increased by deficiency of phosphorus. Another example is the soybean aphid on soybeans. It has been shown that these aphids produced 39% more nymphs potassium deficient than on healthy plants. A possible reason for this phenomenon is that potassium deficient soybeans contain a greater amount of amino acids which are required for optimum growth of the aphid. So healthy plants are more resistant to the aphids than those without soil nutrients. Short generation time, high fecundity. Biological factors promote resistance. Short generation time, high fecundity. No or little migration occurs between populations. The species is highly mobile, increasing the possibility of exposure to insecticides. Biochemical mechanisms of resistance. The principal biochemical mechanisms of resistance to insecticide involve their modified, less sensitive cholinesterase action, glutathione S transferase action, or cytochrome P450 dependent monooxygenation. Both quantitative and qualitative differences in cytochrome P450 isozymes are under general control and both are related to resistance. Recent characterization studies involved ligand binding and multiplicity of isosomes in Musca domestica or the housefly are discussed in relation to resistance. The recent demonstration that multiple isozymes of glutathione S transferase exist in susceptible and resistant insects is of interest, and summary examination of their role in the mechanism of resistance is required. Similar enzymes, but without pyrethroid esterase activity, is also found in the housefly. The resistant such enzymes may serve either to catalyze hydrolysis or as binding proteins. It has been suggested that from time to time that regulator genes, enzymes induction, and gene amplification all play a part in controlling biochemical mechanisms of resistance. Through clearly defined evidence has not always been sought for. Environment, insecticide, penetration, excretion, degradation, interaction, resistant insect. Behavioral modifications, insecticide reduced penetration, increased excretion, increased detoxification, target modification. Isozymes of glutathione S transferase.